Yeah, so we're going to go through all these levels, and I will always draw this line with, okay, how these different elements of within the tissues are changing aspects of mechanical properties of this tissue. So looking at the landscape of the central nervous system from a macro to a nano perspective. So of course, at the macro scale, we have an anatomical division. We can look at different regions of the brain. But if we zoom in a little bit, then if we go to micrometer range from 10 to a few hundred micrometers, we'll see the different cell types within the brain composing this tissue. And this includes not only the neurons. So we know the human brain has an estimated 107 billion cells, but these are not only neurons, only a bit more than half of these cells are expected to be neurons. The other half, or a little bit less than the other half, then are composed by these glial cells, so microglia, astrocytes, and oligodendrocytes. Yeah, that's all right. If you can hear it. And if you can. Okay. So, I think let's see. Okay, great. Yes, so I want to bring from the cellular perspective which cells are composing, what are their functions, and what we know about their contribution to tissue mechanics. Then if we zoom in further a little bit more and look at a few hundreds of nanometers, we can see the functional units of the brain, so the synapses, and I know most of you perhaps are coming from more of an engineering perspective, and if you have questions about this, please feel free to ask. But here we can see close by that the synapses, so this communication between neurons, this functional unit of the brain, is formed by a presynaptic terminal, which in mechanical synapse is composed of the synaptic vesicles that has the transmitters that are going to activate the postsynaptic membrane. And what we're going to see is that this is not a communication only between two neurons. We have as well the participation of astrocytes, so other cell types there, and as well as of non-cellular components such as the extracellular matrix. And if we were to look even further and zoom in even further within the tissue, we could even appreciate how this is the interaction between cellular and non-cellular components, so how the extracellular matrix is binding to the cell membrane. So starting with a microscopic perspective, and of course we have this anatomical division. We can clearly differentiate the left from the right hemispheres. We have anatomical division of the lobes, so we have a frontal lobe, parietal lobe, occipital, temporal, the cerebellum, and this is somewhat also associated with the function of these structures. But if we cut open the brain and look at it, we can see very distinctively a contrast. The outer part of the brain, here lining the cerebral cortex, it's darker, and we call it the gray matter, and inside of it we have a pale substance, almost whitish, called the white matter. And what are these substances and what is their structural organization? So here we have another section of a human brain where you can appreciate the neurons that have been stained and in the purple color, And here again, you see that the outer part, so the cerebral cortex, which has about 2.7 to 3 millimeters of thickness, are way darker and dense compared to the white matter, where you have this pale staining and even other areas without any stains. So what are these structures within the gray matter, which you can see here based on this staining? We have a cell dense region. So a lot of neuronal bodies are sitting here, and their projections are then composing the white matter. So the region where you have these bundles or clusters of myelinated axons, they are transmitting information to other brain regions and also to the spinal cord. From this, we can already appreciate how the micro scale is being defined by the structure at the micro scale. We also know that this has a reflect in mechanical properties of tissues, with the average gray matter stiffness being lower than the average of the white matter tissue. But not only that is relevant, we see also that there is a regional differences in stiffness in the brain, with the frontal lobe being stiffer than the cerebellum, for example. And to try to understand that, we can also use mouse models and other animals models as well. Here you see a sagittal cut of a mouse brain, where sustained for nuclei, the white matter here with the myelin basic protein staining for neurons. So you appreciate you have gray and white matter. And just as I showed you for the human brain, we have shown using MRE, so magnetic resonance elastography, as Inko presented, that the cerebellum, so the posterior part of the brain, is softer than the anterior part of the brain, in the cerebrum. And to try to really dissect what's happening there, it's not just based on the content of gray and white matter. We have to take into the consideration the complexity. So we know, for example, the cerebellum in a mouse has about 10 times more neuronal density than the cerebral cortex. So this could also be explaining the softening compared to the rest of the brain. But let's look at other cell types. How do they influence stiffness? So again, here you have a section of a human brain. And what if we could actually take just a piece of a very small piece of this and really dissect the different cell types and networks within this region? This has very beautifully been done and published last year based on electron microscopy data, where all the cells were segmented and reconstructed within 1 cubic millimeter of brain tissue from the cortex. And here we can appreciate the diversity of neurons. So they're actually not always the same. We have different morphologies and size of neurons, but we also have a very intricate network of vasculature, so which are composed by endothelial cells and also pericytes laying outside these endothelial cells. Besides the neurons and vasculature, as I mentioned before, it's very important to consider the glial cells. And these are different types of cells. We have astrocytes, which are regulating synaptic activity and also giving support to the neurons functionally. Oligodendrocytes are the cells producing the myelin that forms the white matter. Microglia are the immune surveilling cells in the brain, so they're reactive against pathogen. They're always trying to phagocyte whatever must be excreted, let's say, from the brain. And of course, despite being a gray matter region, we see still myelinated tracts because they're then stemming from these neuronal bodies and being condensed or more concentrated within the white matter. Besides the cellular diversity that we can appreciate here, we can also consider that there is a very unique and heterogeneous extracellular matrix in the brain. And these are the components that I want to discuss with you today. So starting with the neurons, despite their morphological diversity, we could say that there is, let's say, an anatomy of a neuron. The cells have a cell body, or soma as it's called, where it lays the nuclei, most of the organelles of the cell. And from this cell body, we have projections called dendrites that forms these dendritic trees, which varies a lot from different types of neurons. Within the dendrites, we also have these very little dots, the dendritic spines. So these are also regions between the contact or forming synapses with other neurons. They are receiving input areas. And one of the projections is often specialized and becomes an axon, which can be myelinated or not, depending on the distance where it needs to travel and send information. But it is the dendrite, let's say, that it's specialized and will then communicate to the next cell, or it's an output to give information transmitted to the next neuronal cell. I briefly mentioned that there is a great variety in morphology. This is true. You can see here from different neurons an example of a cortical neuron, a hippocampal neuron, and a neuron from a dorsal root ganglia, so the peripheral nerve system. And they are not only different morphologically and in terms of size, also of the soma, but they have different stiffness properties. So the cortical neuron is softer than the hippocampal neuron, which is then softer also than the Bioenginyeria neuron. So this may also be contributing to local stiffness properties. We also need to consider, as I mentioned, other cell types, such as the glial cells, and two of the main types are astrocytes and microglia. Astrocytes are participating very actively within the synapses. They're also important during developmental maturation and elimination of synapses. Within the synaptic space, they also contribute to ion and neurotransmitter homeostasis. And they have a very specialized projection that also goes in contact with the vessel walls, regulating blood-brain barrier function and integrity. So basically regulating what can be exchanged between the blood and the brain. The microglia are the cells, as I mentioned, they are immune cells of the brain, so they're constantly surveilling and looking for things that need to be phagocyted and pathogens. They're very responsive to inflammation as well, and regulate synapses via modulation of the extracellular environment, as we'll see more in a few slides. So what about these cells? How do they contribute to the mechanical properties? So we know actually that there is a difference between the stiffness of the cell and the single cell level depending on their origin. The astrocytes that come from the gray matter are stiffer than the astrocytes coming from the white matter. And the opposite is observed for microglia. They were shown to be softer in the gray matter than in the white matter. So in one study, the cells were extracted, microglia cells were extracted either from gray or white matter and cultured. And here you can see again that the average elasticity values for these cells is lower for those that came from the white matter. More important than the origin, it's also knowing their activation status. So if the cells are surveilling all the time, they're looking for pathogens, they're also phagocyting all the time. And this leads to changes in their morphology and therefore also their mechanical properties. So here, I hope you can see, it's a bit difficult to read, but these cells were cultured, so microglia coming from white or gray matter were cultured under three conditions. One is an inflammatory condition with LPS, another one only with myelin debris that would have been simply phagocyted by these cells. Then a mixture of LPS and myelin, which is mimicking neurological conditions such as multiple sclerosis. And we can see that the response of these cells to either of these stimulants is quite different. So the microglia within the white matter becomes softer upon inflammation, whereas the microglia from the gray matter becomes stiffer during their inflammation, at least in culture. In the presence of myelin, both microglia respond similarly with a reduced elasticity, And in response to myelin and LPS, the microglia within the white matter becomes less elastic, and it's unchanged on average for the microglia from the gray matter. So the message here is that we have to consider which cells are present within the tissue, where they originally came from, so whether it's a white or gray matter cell, and the activation status of these cells. Then we also need to consider the vascular network within the brain across different regions. Here you can appreciate the whole vasculature of a mouse brain up until the capillary levels. And what we can clearly see here is that the vascular density from regions such as the cerebellum, cortex, are much higher than the density within regions such as the hippocampus and striatum. And this, of course, is associated with the energetic demand from these regions, but it must be taken into consideration. And what is the effect of vasculature within the tissue and to the mechanical properties? So a very nice study conducted by Mack has shown by pairing magnetic resonance elastography with arteriospleen labeling and quantification of cerebral blood flow, that deep gray matter structures are nearly correlating with the flow rate that was calculated from the cerebral blood flow. So that means stiffness was being correlated with perfusion within the brain. So the more perfused regions were actually stiffer. In another study, also confirming this, by induced hypercapnia, also leading to an increase in perfusion within the brain. It was shown with magnetic resonance elastography that the brain gets stiffer compared to baseline or in the recovery phase. So
we should comment here, perfusion could mean the volume of the blood or the fluid which is pressed through the tissue, or the pressure. And this correlates with the pressure. So if you normalize the volume, which is measured here in the CBN as cerebral blood flow, which is just the volume of fluid perfused through the tissue, you have to normalize this by the diameter, the mean diameter of the vessel, to come to a kind of pressure. And just this normalization brings this linear correlation. So it's not correlated with the volume, but with the perfusion pressure, which is more into the biomechanics and Hooke's law compressive stretch of the vessel walls, which gives an overall increase of the nonlinear shear module. Sorry.
No, thank you. Yes, please, super. So the important message here also is that we have to consider vascular parameters as well for the brain. Another and it's its object of research, and another important point is pathological states. So how does pathology, and in our case, we investigate inflammation, changes the cytoarchitecture of the tissue, and then the mechanical properties. So here you can see a multiplex fluorescence of mouse brain. So the first panel, it has this region around the third ventricle, and here you have a bit of white matter and the thalamic region. So here it's flipped, but we can appreciate the main types of cells within this region. So we have the neurons in magenta, the microglia are being labeled in white, which here is very difficult to see, but you can trust me, it's there. We have astrocytes in green, nuclei in blue, endothelial cells or vessels labeled in yellow. What happens during autoimmune neuroinflammation? So using a model of multiple sclerosis where we actively lead to a massive breakdown of the blood-brain barrier and infiltration of leukocytes in the brain, the picture of its cytoarchitecture changes completely. So you can see here that the leukocytes that are often not observed in the brain are now there in cyan, leading to a massive inflammatory response with reactive astrocytes and general gliosis, so microgliosis and astrogliosis. And the result of this, as we see with MRE, is that the brain of these mice with autoimmune inflammation is softer than their brains prior to the induction or compared to healthy animals. Still looking from the perspective of pathologies, so multiple sclerosis has on one hand this inflammatory feature, but it is also a very demyelinating disease, so it's basically known as a demyelinating disease. And the degree of myelination, it's also associated with mechanical changes, especially within the white matter. So here using a model of toxic demyelination, that means animals are fed with cuprizone that causes death of oligodendrocytes. So if you remember the cells I told you produce the myelin, leads to a progressive loss of myelin, as you can see here with the myelin staining. But you can also measure this with MRI. So titulated images, this corpus callosum, which is a white matter region that is connecting both hemispheres, is seen as this hyperintense area. So it's very dark. But over the course of this demyelination, we don't see this region anymore, so it becomes more hyperintense. And this is also associated to some degree with reactive astrocytes. So in a previous work, also from the group of Professor Zach, the stiffness was measured within this white matter before and after demyelination. And we can see that after 12 weeks of feeding these animals with cuprizone, there is a loss of stiffness within this region, here confirmed with another type of myelin staining. So here we could say, okay, there is also astrogliosis, but there are also other studies in which the degree of myelin is increasing. So for example, with maturation of the brain, where we see that this increase in myelination leads to a stiffening of the brain. So we have different models used for detection of the degree of myelin and how this affects local stiffness. Then we need to consider the extracellular matrix as well, because the matrix in the brain, it's very unique in the sense that it's compartmentalized, so it's very heterogeneous, and it's also highly functional. It's not just there to give support to the cells, but it has a functional both in physiological processes, but also pathological processes. So the matrix in the brain, Ingo presented in his talk in the beginning, it's really reaching glycosaminoglycans. So proteoglycans are these core proteins that have the side chains of glycosaminoglycans, and it's a very soft matrix. Its compartmentalization depends on the region. So, for example, around the cell bodies of neurons, we find what we call perineuronal nets. So, basically, like mesh-like structures made of chondroitin sulfate, proteoglycans, hyaluronic acid, and some binding proteins. And these are very important for regulation of synaptic activity, as I'm going to show in a bit. The basement membrane, so the matrix that is surrounding the vessel walls, and it's being produced both by endothelial cells and the astrocytes. It's more fibrous. It has collagens, laminins, fibronectin, and some heparan sulfate proteoglycans. The interstitial matrix is a bit of a mixture of these two. It's very rich in chondroitin sulfate, but it also contains to some degree specific types of collagens. And we have a matrix between the nodes, the myelin sheets, and this is mostly also based on chondroitin sulfate and other proteins such as kinase. So let's look at have a closer look at the perineuronal nets and see how they were associated with changes in stiffness and also pathological changes. So perineuronal nets are often visualized with a lectin from plant lectin called WS1. These bind or intercalates with N-acetylgalactosamine moieties from the glycosaminoglycans, and we can see generally the delineation of the neuronal bodies and a bit on the dendrites if we zoom in. So here from the mouse brain, you can see that the cortex is a very rich, PNN-rich region. There's also a high concentration of PNNs within the hippocampus of the mouse brain. And that's very, that makes completely sense because these are the regions responsible for cognitive function. And these perineural nets are responsible for neuroplasticity or at least preventing neuroplasticity. So if we zoom in a bit further and using super resolution microscopy, we can see this mesh-like structure of the PNNs, and they're basically visualizing the glycosaminoglycan chains of the chondroitin sulfate proteoglycan. How does these perineural nets are regulating then plasticity, and what does that mean in pathological states? So first, here we can have a look at the neuron that's being stained with MAC2. So you see the whole projections, the WFA in red showing the perineural nets. So you see it extends also through the dendrites. And when we stain for synapses using synapsing, that we are able to visualize presynaptic terminals, we can see there's the co-localization, and the WFA is actually either co-localizing or in the vicinity of these structures. So the idea here is that the perineural nets are actually stabilizing already existing connections. So you have the pre and postsynaptic terminal, and the perineural nets are stabilizing these two components of the synapses, and also preventing neuroplasticity in the sense that they are creating a physical barrier that does not allow the contact of an axon terminal with existing dendrites within the neuron or via chemical repulsion. So what happens during the autoimmune neuroinflammation, so the case that I brought to you before, we see that in the cortex of these animals that inflammation leads to a drastic reduction in the intensity of these perineural nets, indicating that they're somehow degraded. And because the disease, it's a relaxing remaining, so it has a recovery phase, we can see later on that this perineural nets are then restored. And we quantify this here, as you can see, the signal intensity of the perineural nets. And when using MRE to detect the stiffness and quantify the stiffness of this tissue, what we saw, to our surprise, was a very similar pattern. We have a softening during the inflammatory phase and a re-stiffening, let's say, to its basic stiffness at the remission phase. We correlated these two parameters, and they correlate also very well with disease activity. So that suggested to us that the perineural nets are also relevant in bringing mechanical stability to the cortex. And when they are degraded, this is lost, leading to tissue softening. And that can be used also as an imaging marker of disease activity and inflammatory activity in the brain. Finally, at the nanoscale, so it's not really nanoscale of a few nanometers, but maybe hundreds of nanometers, we can zoom in into this functional unit of the brain and have a closer look at the synapses, at the dendrites, and in the dendritic spines. And here, most of the techniques applied are based on super resolution microscopy. And what we can see here very nicely is the interaction between astrocytes and neurons at the dendritic spines. And the upper figure here you see in green the neurons, and in red the astrocytes, and the areas in blue are the sites of interaction between the two. And this is very relevant for understanding physiological processes and also in neurological conditions, how this is occurring. In the panel below, we see the opposite of the colors, and now the neurons are in red, and then the astrocytes are in green. So also showing that they are in close contact and interacting. Another possibility is that super resolution is used actually for the quantification of the dendritic spine. So the density or numbers, something that's not possible, for example, here. In two-photon microscopy, two dendritic spines are seen as one, but they're well resolved using two photon coupled with SED. And this is very important in pathology. For example, in Alzheimer's disease, we know that these dendritic spines are retracted. They are reduced in models of Alzheimer's disease. And this gives us a tool to better quantify and visualize these processes. If we then look very closely at one of these spines, it's also possible to understand better mechanisms of diseases. So here, for instance, in this study, F-actin filaments within one dendritic spine were analyzed in wild-type animals and animals with a mutation that leads to the Alzheimer's or a model of Alzheimer's disease. And they found that the structuring of these filaments is completely changed in the animal model, with a reduction of the filaments, completely different rearrangement, and depolymerization of these actin filaments, which may be explaining how, at the end, in Alzheimer's disease, there is a reduction in these dendritic spines in the dendrites. So, of course, the possibilities at this scale are various. We can understand better the density of receptors in the postsynaptic terminal, the synaptic vesicles within the presynaptic terminal, the interactions with the matrix and other cells. But what we don't know yet is how, at this scale, at this level, these subcellular structures are interfering or contributing to mechanical properties of the cell and the microenvironment. So to give you a summary, I hope you could have an overview of this hierarchical structuring of the brain from the nano to the microscale. Here at the nanoscale, we saw subcellular structures, so the synapses, dendritic spines, and filaments, and how we can have a closer look and investigate this in pathology and physiology. Later on, we also saw at the microscale, or before that, how the cytoarchitecture is important for stiffness of the tissue. So not only the density of these cells, but the activation status of the cells, which is also being present in the tissue. So the composition of neuron glial cells, vasculature, and also of the extracellular matrix. And finally, that this, I hope, it's somewhat clear, has then an effect at the microscale at the mechanical properties of the different brain regions. So, of course, here we saw how structuring of the gray and white matter leads to different stiffness within the different regions, but also as a reflection of this macro scale. The challenge that remains, and maybe some of you are going to be tackling during your PhD, is how to integrate all these levels, so from the nano to the macro scale, and perhaps create predictive and multi-scale models for understanding these tissues, if not in the brain, then with other tissue types and on other models as well. Thank you. I'm happy to take any questions that you may have, and I hope it was not too much biological.
I have a question. Let's say that we want to measure the stiffness of neurons now. For example, with an AFM. We cannot use staining techniques. So how can I discriminate the neurons and the astrocytes or the other components? And if I cannot, I mean, the measurements can be considered reliable. So how much is strong the influence the other components?
Yeah. So one option is that you isolate neurons and culture them, and you can have a cell culture of neurons. If you're meaning within the tissues, then you have alternative ways of doing that. So you're not staining the tissue because then it's going to be dead, right? But you can use reporter mice. So, for example, there are mice that have green fluorescence protein exclusively present in populations of neurons or astrocytes. And if you have your system coupled to fluorescence microscopy, then you could just select the neurons that you like to measure.
So is it possible to culture humans without anything else?
Yeah, of course. Yeah, so many of these results for stiffness of single cells of neurons and microglia, they're all coming from AFM and also from cell cultures. So this is possible. And within tissue, I would say using reporter mice.
Thank you.
I just had a question about the experiments with the animals. Like they're being fed some compound which leads to demyelination. Does that mean that all the myelin from everywhere goes away?
To some degree, yes.
So like they can't move anymore, eat.
No, they can.
Muscles.
Yeah. This would be a
How do
logical.
they survive?
They do survive. They don't actually have many symptoms that we can detect. For example, motor symptoms. The animals are still moving fine. We have some degree of demyelination. It's not 100%, but the animals are still functional. And this is not a specific, of course. So as you mentioned, we're killing the oligodendrocytes everywhere. So you have demyelination in the spinal cord, demyelination in the brain. Just the easiest way of seeing demyelination in the brain is looking at the corpus callosum, but you would expect that this happens to a certain degree everywhere. But it's not 100% of demyelination.
Like,
so we have some
systemic effects not have any, like if the myelin from everywhere is gone, right? Like the spine and everything, would that also not affect reflexes and motor movement?
Yeah. So perhaps with behavioral studies, these could be detected, but we don't see so clearly any functional differences within these animals. So maybe the degree of demyelination is not that strong that would lead to completely, let's say, failure of the central nervous system, like a shutdown and dysfunction.
I have a question for the showed some data on cells that are derived from white and gray matter, so differences in the mechanical properties. So I wonder, when you take out these cells, because we have these basic changes in the tissue mechanics, do you think it's like adaptation of the cells, or would that persist if you culture them, or is it these cells were freshly derived and then measured?
The experiment that I showed was with primary cell cultures. So they were extracted from gray and white matter and plated. Then, of course, we don't know how much physiological is really these cells are behaving compared to with these cells in their natural environment. But yeah, this could also have an effect. So how we plate them, what kind of substrate, for how long they were plated, and maybe passages, all these factors must be taken into account. This could be altering also their behavior. But it seems that these cells naturally, from their origin and probably the stiffness within these regions, they are acting differently. One hypothesis is that also actin filaments are differently regulated between the glia cells from the gray matter and from the white matter. But I'm not so certain this is really accepted as the final mechanism that explains why they are so soft.
You showed these data with huge differences, right, between the different types of neurons. So in the order of kilopascal, reliable is it?
I am not sure. I must say that. I know these are also coming from AFM measurements, but I'm not sure how much this has been reproduced with these specific cell types.
Single publication?
I think these values were from only one publication, but I think it's generally accepted that then the morphology, they also have completely different size in soma, might have an effect, but I'm not sure how much reproducible is that, I must say.
No, how long they were cultured?
No, sorry.
Or not substrate?
No, sorry. Yes, I think that was a bit, but I can check for you and we can talk later. Yeah, but yeah, it does make sense. Yes.
I have one question about the clinical properties measurements, especially stiffness. are all the data that you got from SPM systems, or do you have some non-contact stiffness measurement techniques?
So my data were only the ones with the mouse model of multiple sclerosis, and this is all MRE. Then also the data with the humans and perfusion were MRE based. There are also some confirmation with ultrasound, but we don't use SPM, and I also haven't worked with AFM as well.
Okay, do you have an idea about how great are measurements, stick stimulations or things like that, compared to contact measurements like Or just more question, are you more interested on qualitative measurements comparison, or qualitative, you really want to know the stiffness?
Yeah, this is really good. We always have to compare, right? It's not that we're going to reach a single value of stiffness per region and say, okay, this is the basics. You always have to compare conditions. So what we're doing, basically working with pathologies, is looking at a healthy versus a sick. And then seeing, okay, based on what we see structurally different within this region, what kind of elements are leading to these changes. So it always has to be comparative. So we're not trying to get any final quantitative value for the regions. I'm not sure about Ingolf, maybe also can.
I mean, I would call it quantitative. So we have shear wave speed in meter per second, or complex shear modulus in kilopascal, but it's a kind of self-consistent bubble of MRE here. I mean, wave-based elastography or ultrasound, which gives values which can be compared. As long as you know the vibration frequency, which we know, which is precisely set in our experiment, right? And it's a mouse brain. We are in the kilohertz range, so we cannot directly compare to the human brain, which I showed this crossover of stiffness values, even relative stiff versus soft can change in the kilohertz range versus the 50 hertz range of the clinical MRE. But if you keep this in mind, you end up here with MRE with quite consistent values. It's, I mean, we have some experience with AFM, and it's easier than AFM, I would say. As you don't need the tissue preparation, so your in vivo, you put the mouse or the volunteers are the easiest subject, by the way. So you put them into the scanner, you shake the brain, and you get a map out, which is quite consistent. So we made repeated experiments after a year. It was a variation of about 0.5%.
One very basic question. So I guess you are recording the frequency response of the tissue. Do you send wave, and you measure the vibration of the tissue in order to reconstruct the stiffness? It's actually better way.
Exactly. So we will have further talks and lectures on that. Yeah, exactly. Therefore, we need the viscoelastic models to come to frequency independent parameters, which can be compared.
Okay, thank you for the questions and discussion, and I hope you enjoy the rest of your workshop.
Genau, ich denke, dass das jetzt das Wichtige ist. Das ist überall. Ich weiß nicht, wenn man das Physik aufmachen, was dann passiert ist. Also, genau, das ist, ich habe Und damals habe ich schon mit der Definition der das soll ja nicht noch aber hatte die Erwähnung rumgeschickt? Die Erwähnung oder?